Good morning, students. So till now we have seen the gametogenesis process as well as insemination, that is nothing but the gamete transfer process. After transfer of the sperm or the male gamete, we have seen that it will finally reach to the egg and the haploid nucleus of both will fuse, and that results in the formation of zygote. With that we have seen the fertilization process. And now it is the time to see what exactly happens after fertilization. That is, what is happening to the zygote. How the zygote will develop into a embryo, and this embryo, a fully developed embryo, is expelled out from a female body, which is called as parturition. And then, since it is a human, that's a mammal, it has to nourish the younger ones, for which it will do the lactation. So all this comes under post-fertilization event. So we'll see the zygote now. So this is our zygote. If you see here the zygote has a diploid nucleus. As I already mentioned, it is diploid nucleus because this is formed from the fusion of two haploid nucleus. One haploid nucleus was from male and another haploid nucleus was from a female. Those two got fused in the fertilization process, resulting in the diploid nucleus. Then, after the nucleus, it is the next membrane called as vitelline membrane. So we know that once the sperm enter into the egg, there is a hard membrane formed around the egg, so that it is preventing the entry of other sperms which are being in contact with the egg, that is polyspermia. So that membrane is called as vitelline membrane. Clear? After that, as usual, it is the next layer called zona pellucida and it is the corona radiata. So this is about the zygote, and one end you can see here, this is the animal pole, and the other end you call it as a vegetative pole. Now it is the cleavage of zygote. The zygote will start dividing, we call it as cleavage. Cleavage always happens after fertilization. So let us take first thing, it is the cleavage of zygote. So the zygote here will undergo division. This is zygote, nucleus, diploid nucleus. You'll have the membrane, the corona radiata cells here. So now there is a division here, resulting in the formation of two cells. The zygote, single cell, has undergone division, resulting in two cells here, and each cell you call it as a blastome here. So now this is a two cell, or you can find two blastome here, and this division is unequal. As a result, you can see one bigger cell and one is a smaller cell, and always the division is from the animal pole to the vegetative pole. That is, cleavage start from here, so then it will move towards the vegetative pole, and all these are still intact. That is, you can also find the zona pellucida and corona radiata, all this are still intact. It is only the inner cell, the zygote, has divided. Clear? Now, one more important thing is the division here is indeterminate. We say it as indeterminate. First thing is division is unequal. As a result, you can see one bigger and one smaller, and it always starts from the animal pole and move towards the vegetative pole. And second important point is division is indeterminate, meaning the fate of these cells are not yet decided. Therefore, each cell can give rise to anything. It can develop into new individual. At this time, we don't know the fate of this one. This has the ability to develop into a new individual, and even this cell can develop into a one more new individual. That is how you can find the twins. That is identical twins. I am talking of identical twins. Each cell here can give rises to different embryo. This is one embryo and another is embryo. So this is the second step. After this further undergo division. So now it results in the 3 blastomere. It was 2, now it is 3. And this 3-cell blastomere stage, it is very short-lived. Immediately it will divide into 4. The first, the large cell which you can see here, this will undergo division to form these 2 cells, and membranes are still intact. Every time I am not going to write, but see here, even the zona pellucida is still here, and along with the corona radiata is also here. But this is a short-lived. And immediately this is going to divide further to form a 4-cell blastomere. Clear? This was a zygote which divided to form 2-cell blastomere, and this was 3-cell, and here it comes 4-cell blastomere. Now here also the membranes are still intact. Continue writing this. Zona pellucida and corona radiata are still here. And then it is from 4, again you can see they further divide to form 8-cell blastomere. From 8-cell to it is 16-cell blastomere, and 16 to it forms into a 32 cell. See, 16-cell blastomere here, it is also called as morula. But during this, that is between this 4, 8, 16, by the time it reached to the morula stage, the corona radiata membrane are lost. Clear? Between this process, the corona radiata are lost. Clear? And now from 32 cell, it is, from 16, it is the 32 cell blastomere, and here you can see that the rearrangement of morula cells will take place. The 16 cell will get rearranged, and then it arranges such that the smaller blastomeres will move towards the peripheral and larger towards the inner. Clear? I'm going to take after morula into a second phase, that is cleavage of zygote. Here what we have seen is the zygote divided and it resulted in the morula. We'll take it the next step, that is the formation of blastula. So now here we'll take this morula. I told you this is a morula, and this morula should have, if I say morula, it should have 16 cell. So there are about 16 cell here, but these 16 cell morula will rearrange themselves such that out of the 16, the smaller blastomeres, 1, 2, 3, 4, 5, 6, 7, 8, 9. So these are the the smaller blastomere. The smaller one you call it as micromere. The smaller blastomere are called as micromere, whereas the larger ones, I have written 12, 13, 14, 15, 16. So now the larger one is called as the macromere. So this is the blastula. Am I clear? The zygote divided to form 2 cell, 3 cell, 4, 4 to 8, 8 to 16 cell, and at this stage you call it as a morula. Now there are 16 cell inside this morula. Out of these 16, the smaller cells will arrange themselves at the peripheral towards the end, and bigger ones will move towards the center. And the small one you call it as micromere, the bigger one you call it as a macromere, but both are the blastomeres. Now it is further it will divide. Further it will divide to form the blastula, that is from 16 cell it will divide to form the 32 cell stage, and that you call it as a blastula. So now again you will find not only 16, we find 32 cells which is called as blastula. During this process, from morula to the blastula, the zona pellucida is lost. Here, the zona pellucida here corona radiata was lost, but still the vitelline membrane and the zona pellucida was there. But when it comes to the blastula, even the zona pellucida is lost, or you can say it is broken, and then it is this. See here, morula will actually reach the uterus. We need female reproductive tract. 1 second. Yes, you can see here, this is actually a ovum. Now it is that the sperm has fused with the egg, resulting in the, after fusion it results into the zygote. After that, the zygote, which was single cell, will divide to form 2 cell, then it is a 4, from 4 to 8, 8 to then it is the 16 cell like this. At 16 cell you call it as a morula, and all these events will happen in the fallopian tube. If you remember, I told you ova, that is after ovulation, when the ova are released, it will stay in the ampulla region. After fertilization, whatever we have seen here, these events happen in the fallopian tube. Once it reach to the morula stage, these morula leave the fallopian tube and come to the uterus, leaving the fallopian tube. Leaving this fallopian tube, it will come to the uterus. It is here the morula will be next converted into the blastomere, and the blastomere will attach to this uterine wall. So let us now see how the blastula will attach to the uterine. As soon as the endometrium layer, see, we know that when the blastula go and touch this uterine layer, the endometrium layer which is there, which is the innermost one, where is that? Yeah, this endometrium lining which you can see here, this contain the glandular cells. They secrete the nutrition material. This nutritions, suppose if this is an endometrium here, they secrete the nutrition. These nutrition are absorbed by the blastocyst, and this will make it to increase the size of the blastocyst. Clear? At the same time, the trophoblast, see here, I forgot to tell you one more thing, and this is a 32 cell stage, you call it as blastula, right? So the outer region here, the smaller micromeres which are there, they are converted and they are formed into the trophoblast. Clear? The micromeres form the trophoplast. Trophoplast is formed from micromere, whereas the macromere which was present, the larger cell which was present at the center here, they will be called as inner mass cells. Inner mass cells. Hope I am clear. Micromere will be converted into trophoplast, and the macromeres which are present, they form the inner mass cells. And now it is this blastula. So this is from morula. It has been converted into blastocyst. The outer region here, outer region we called it as trophoblast, and inner mass you called it as the inner mass cells. Now the endometrium layer which secrete the glandular cells, the endometrium layer contain the glandular cells. These glandular cells will secrete the nutritions which are absorbed by this blastocyst. Once it absorbs the nutrition, even the size of this will increase. It grows into a bigger because nutrition has been absorbed. And so same here I am going to write here. It has increased the size. This outer layer we called it as the trophoblast. And at the same time, even the trophoblast, they will secrete the enzymes to loosen the inner mass cells. And what is the space here is, here the nutrition was absorbed. I told you the glandular cells which secrete the nutritions have been absorbed by this blastula. Therefore, those nutrition which are absorbed are stored here. Since it needs a space to store, it will push this inner mass cells towards one side. That is why you can see inner mass cells are being at one side, and here you can find a small cavity, and this cavity you call it as blastocele. Blastocele, and inner mass cells as they are pushed towards one side. Now our blastocyst is ready. This is our blastula. This blastula here will now go and attach to the uterine valve, and that is our next event called implantation. Yeah. Next is implantation. See, once here I told you this will be the morula. Now morula has been converted into the blastula. That is 32 cells. Out of 32, they are like you can find the outer trophoblast and inner mass cells. Apart from that, they can also see a cavity. There is a cavity called blastocele. And now this blastocyst is now ready to get attached to the uterine wall. The attachment of the blastocyst into the uterine endometrium, we called it as implantation. Let us see how this implantation happen. See, as soon as this trophoblast, just one second. See, as soon as this trophoblast come in contact with the endometrium, hope you know this innermost you called it as endometrium. Endometrium, the middle layer here you called it as, the middle layer is called as myometrium, the outermost you called it as perimetrium. Now as soon as the trophoblast come in contact with the uterine wall here, it is that the blood vessels which are present here. So this is the blood vessel, this is a blood vessel here, and this blood vessels in the endometrium layer, they increases their permeability. They can easily move because they have increased the permeability. And apart from that, the fibroblast, that is, if I write here, that's a fibroblast which are present inside the uterine wall. They are converted into glycogen filled cells. Filled cell. And this will provide the nutrition. This is one side preparation. On the other side, what happens? The blastocyst will have the trophoblast. This trophoblast will start producing a finger-like projection outside, and these are called as chorionic villi. The trophoblast produce a finger-like projections which are called as chorionic villi. And also the endometrium lining, what you see here, they will get rupture and they start growing like this. The endometrium get ruptured and chorionic villi appears on the trophoblast, and they penetrate till they come in contact with the maternal blood. Maternal meaning mother's blood. The chorionic villi will come in contact with this mother blood, and apart from that, even the endometrium layer which is there, you can see it has now covered this blastocyst, and this you call it as implantation. Now what happens here is the chorionic villi and this endometrium lining forms a single lining. They will merge and they form a single layer, and now this you call it as the placenta. Chorionic villi surrounded by the uterine tissue and the maternal blood. So there is now a temporary association between the fetal tissue. Here the fetal tissue meaning we are talking of the chorionic villi which is coming from the trophoblast. There is an association of this chorionic villi and also the uterine. This endometrium is from mother. Chorionic villi was from the this blastocyst. Therefore, these two will unite together to form the placenta. And now the placenta will give the nourishment. Now the role of placenta is also important. They can ask you what are the functions of placenta. Functions of placenta. Yeah. So these are the functions of placenta. And one more thing here is if this is a placenta and this is our blastocyst, clear, this will be our blastocyst which will further develop into an embryo. There is a tube which is coming from placenta to this embryo. This one you call it as umbilical cord. The embryo, this is our embryo. See here, I'll write it in a different way. Okay. This is our placenta. Clear? So here is our umbilical cord. And then this is a embryo. And this is the one which is going to develop into a baby. This one. This is our embryo. And this is the placenta. You can see a connection here which is coming from the placenta and it connects to the embryo. Coming from the placenta which connects to embryo, this one you call it as, I'll write here, it is called as umbilical cord. Umbilical cord. Okay, it's a connection between a embryo and the placenta. Once the embryo is expelled, they cut this umbilical cord. Now, what is the function of the placenta? So here the placenta will actually transport the oxygen and nutrition from mother to the embryo. This growing embryo needs the oxygen and also it needs the nutrition. Both are taken from the mother. Then it is given to this embryo, and that work is done by the placenta. First thing. And second thing, simultaneously the carbon dioxide which is formed the carbon dioxide, and the waste which is formed from the embryo are removed from the embryo. This will be the second point. Apart from this, they will act as a endocrine glands, secreting certain hormones like human chorionic gonadotropin, human placental lactogen, estrogen, progesterone. So these are all the hormones secreted from the placenta. So this can also be an important question. Name the hormones secreted from the placenta. Clear? So these are the functions of placenta. Hope I'm clear to you all. Clear? The trophoblast which will produce a finger-like projections are called chorionic villi. These are the chorionic villi, and here the endometrium layer of a female will cover this blastocyst, and now both this endometrium lining and the chorionic villi will fuse together and it will act as a single unit, and that is called as placenta. Placenta is a connection between a mother and a fetus. Umbilical cord is a connection from placenta to the fetus. If they ask you, the connection between a mother and fetus is always the placenta. From the placenta you can see the embryo are connected by the umbilical cord. Clear? And these are the role of the placenta. With this we have completed the implantation, and still the blastocyst which has been implanted will now further develop into a embryo. We'll take up that in the next video. Hope that you are all getting, and if you have any doubts you can message to my number with your doubts, and please comment whether you are understanding or you want me to explain very less, because these are all the extra things. See, whatever we have discussed here, these are all not necessary for your exam purpose. For your exam purpose, you can just write it simply that the zygote will start cleavage. So the zygote will divide to form the blastomere. It will divide to the morula and then to the blastocyst, and this blastocyst will go and attach to the uterine wall. This much is sufficient. Within 2 lines you can complete. Just comment whether you need all this extra things or you want me to do only for exam purpose. Clear? The next class will take up with the embryonic development and then the parturition and lactation as well. Thank you.